want to start asking questions off the study guide?
I know she has them. I know she has them.
Help me now. This is the time.
Okay, bacterial replication versus eukaryotic mitosis. What are the
Okay,
differences?
bacteria, binary fission. Carrier, either mitosis or meiosis. Most of the microbial ones are mitosis, asexual. There is some sexual, particularly when you start to get up into some types of fungi. But bacteria, for our purposes, only that. So, and we are recording again. That's fine. Anything else?
We have a lot.
Keep going. I'm listening. I'm here. I'm going to do it this way, so I can find the test on my thing.
What are the temp categories, especially thermo?
Okay, the temperature categories, the ones that you need to know. You don't worry about the extremes. Psychrotrophs, roughly 5 to maybe 25 to 30. Mesophiles, 25 to 45. Thermophiles, roughly 45 to 70. Okay?
What are plasmids? What are they made of, and what do they do?
What are plasmids? What are they made of? What are they? Hold on. Yeah, here it is. I got it. Yay. Okay. Plasmids are small circles of non-essential DNA. Useful because they have resistance or the ability to make a capsule. And non-essential means they won't die without them. So essential means it would die if it wasn't there. So, and they can come and go, but they typically have important information that helps them survive, particularly under adversities. More questions before I take a quick look.
Let's see at what.
The test here.
Do you like tell us?
I did. The nucleolus is where ribosomal RNA is made. If you nucleolus, remember that? Ribosomal, that's a guaranteed question. Okay, guaranteed, lock. Algae have chlorophyll. There's 2. Don't say I didn't give you anything.
Okay.
Don't tell the people who aren't here. Don't tell Grace.
So, what are flagella? What are their primary functions?
Flagella. What are the primary? They move. Basically, they're the means for microbes to move around. Again, flagella are much more common in prokaryotes. Eukaryotes, the protozoans utilize flagella as a means of getting around. We just have one, the sperm cell. And they're different. Those are much larger in eukaryotes. They're made of microtubules, that 9+2 pattern, where in prokaryote bacteria, read that, flagella.
And then what about Fimbria and Pili?
Fimbria and pili are smaller still, and they're only in microorganisms, but bacteria. They're prokaryotic. So where a flagella might be this long, a pili might be this long, it's made out of something called pilin. The one that we deal a lot with is the so-called sex or fertility pilus, which is hollow and DNA moves from. They erroneously thought it was some type of male-female, but there's no gender in bacteria. It's all asexual. It's just material being transferred. And then Fimbriae are smaller still. They're probably still made out of something similar to pilin, but that doesn't matter. I won't ask you about that. But that's how they attach to receptors in your gut, on your skin, wherever they go. Generally urinary, respiratory. Okay. More.
So from chapter 1, it has the what are the domain, kingdom, genus, and species. What do they represent?
Genus, not genius.
Like, what do you want?
Bacteria are not going to a Mensa meeting. That's a meeting for geniuses. I didn't get into that.
So we're not. Is it going to be on the test?
What? Domains. The domain theory is what we practice now. It is based on ribosomal RNA and it's based on DNA strands. So all about molecular biology. There are 2 prokaryotic domains, archaea and bacteria. Very similar, but not the same. Bacteria have peptidoglycan, archaea don't, so their cell wall's different. They're roughly the same size. The other way they differ is where they live. Archaea only live in extreme environments. Bacteria kind of live in. That's one. There are, for microbes, there's one big eukaryotic domain, like our cells, but not us. Animals and plants are by definition macroscopic. So you have protozoans, single cell, no cell wall, live in water and soil. You have mold and yeast, essentially, because fungi are pretty much macroscopic. They have cell walls, but no chlorophyll. And remember, by definition, they're saprobes. They live on dead stuff. Still disease causers, and very common, mind you. And protozoans are disease causers too. And then lastly, the ones that have a cell wall and chlorophyll are algae. They're more like microscopic plants. They're water-based, where mold and yeast are basically land-based. Fungi are certainly land-based. And the fungi have that thing called chitin, and probably isn't that relevant until you get to this, or probably until you could see like a tiny mushroom. Still go. I'm still here. I haven't left yet.
Does anyone else have questions?
You want me to do? I'll do the generic stuff. So here, I'll give it to you again. You listening out there? Pasteur was all about germ theory. He wanted to disprove that whole, spontaneous generation. Boom, table comes alive. This is not some sci-fi movie like that. That's important. The distinction, Pasteur was more clinical. Koch was more the lab guy. Okay, with things like that. So vaccines for Pasteur, Koch, augers, isolating things in pure culture, like you're doing in the lab. You're doing Koch techniques. Genetic engineering, DNA transfer is what that's about, using plasmids, because you can splice it. Typically E. coli, also yeast, because they're pretty amenable to that type of thing. Resurgent versus emergent diseases. Emergent, mutations, going into areas we haven't been to, because we can recognize them. It doesn't mean they're new. It means that they're new to us. And then resurgent, worldwide travel, aging, vaccination laxity, immunosuppressant drugs, HIV, the onset of HIV and immunological diseases. That allowed tuberculosis to flourish after we almost eradicated it. Vaccinations are more about measles. It's like we're starting to see people they've got. Somebody told me in my other class today, she was giving a vaccine for the Gardasil, which is for HPV, which may or may not have. I'm not any judgmental at all. When she told me, a 12-year-old girl, and they know they faint typically when they have that, which I didn't know, and so they have them stay at the facility where she was working. They asked the mom or the dad 30 minutes, just let them sit here because mom said she'll be okay. Out in the hall, boom, fainted. And that doesn't mean not get it, but that makes people distrust. Why would it make you faint? Well, immunological things are fun. Okay, peptidoglycan, only in bacteria, nowhere else. That's the word unique. That's part of it. Remember what viruses are. Everybody can get a virus. E. coli get a virus, you can get a virus. Not the same, it's the same. So it affects all, and the big difference, there are acellular forms, viruses, DNA, or RNA, never both, protein shell at a minimum. Prions, proteins only. So that the whole endospore thing, adverse conditions, not enough nutrients, not enough water, not enough carbon and nitrogen compounds. Very few can do it. It takes hours. Remember that animation? Asymmetric. It looks like asymmetric fission, but it's not fission at all because of the two cells. The mother cell has an equal amount of DNA that deteriorates as it envelops the forespore, which is tiny, with the same amount of DNA. So it's just changing from vegetative, growing happy, to the endospore or spore stage. When times get better, whatever that is, more food to eat, better atmospheric conditions, availability of water, they convert back and they start to reproduce just the way they always did, binary fission. So that's a big nugget about it. They're grand. So you have the forespore, you have peptidoglycan in between the membranes, that dipicolinic acid. tracks calcium to harden it, expels water to effectively dehydrate it, and preserve it just the way, again, like we have a food dehydrator. So stuff my wife grows in the summer, she can, all that stuff she can dehydrate and we can use later. Standard stuff, membranes, phospholipid bilayers, nothing unusual about that. The big deal with them is selective permeability. It doesn't include that LPS membrane. That's only on that's the outer membrane on gram negatives, and we call the cell membrane the inner membrane. And the oddball thing about that is it's got phospholipid on the side that's close to the bacteria, but the side that's going away has that lipid A and O-specific polysaccharide or O-specific antigen, whichever is called, because they're the same thing. And that lipid A is unique in the sense that it's only there, that layer, and it's always toxic to us. It's totally foreign to us. So our immune system will respond and sometimes very vigorously. So it will always cause damage in us if there's a sufficient amount of it. The O polysaccharide, we type with that. We know the patterns, we can characterize the patterns, and that way it can tell us, is this friendly E. coli or is this a killer strain of E. coli? So the famous one is O157H7, which you'll encounter down the road. The ribosome is 70S versus 80S in eukaryotic cells, but the mitochondria and the 80S cells, that endosymbiotic theory is important. They have 70S ribosomes. Remember, circular DNA in bacteria, a giant circle that's supercoiled versus our pairs that vary species to species. But our ribosomes and indeed chloroplasts in pigmented organisms like plants have circular DNA, bacterial DNA, and 70S ribosomes, bacterial ribosomes in those energy transforming structures. That's endosymbiotic theory. It's like the precursor cell swallowed a bacteria and said, hey, you could help, make energy. But I'm going to keep you here, be useful. And it did. I'm sure they didn't talk to each other. No one thinks you're funny. That, glycocalyx slime layer, and that's a multicellular, and they're really important today. You see them all the times. They cause a lot, particularly at older. If you have older piping, they're a big problem and things like that. We went to that, those new ones that are like colored plastic. Okay, again, the fertility pilus, it's found, it is hollow, and DNA moves through it. Again, plasmids you already asked me about. They're not incorporated with, normally they can be, but most of the time they're free from, remember it's called a nucleoid. There's no nucleus in a bacteria. It's like there's no crying in baseball. Okay, animal cells and eukaryotic cells all have sterols to help the membrane, even plants. That acts as membrane stability. Fungi is called ergosterol. We spend a lot of time with that. And in our cells, it's cholesterol, which is one of the reasons I'm on keto again. I'm making rolls out of oat fiber. Let's see, vital wheat gluten, not the flour. Some kind of crushed, not hemp seeds, flax seeds, that type of stuff. And are you ready? A lot of almond flour. Okay, the difference, cytoskeleton, very scant in prokaryotes. Ours has microtubules and those intermediate fibers. Those are highly organized. They both have little bits of actin microfilaments. Nucleolus question. You're going to get one right. Maybe two.
How do people typically do on this test?
Like, what's the
Average, not so good to start.
so?
Yes, I'd rather be honest. It's a wake-up call to show the degree of difficulty that you all need to be familiar with going forward. Okay, let me determine what a good grade is. Somebody gets 70%, it's probably going to be a B. I'll scale it accordingly.
So you're gonna curve it.
If I don't, there's going to be, I'll have people threatening me.
Yeah.
Grace with threatening.
You're gonna curve.
I anticipate I'll have to. Unless we have, I mean, I have one student who you work with in nursing now, who's just is the ultimate, she's the best student we've had. I think she's still here.
Is it me?
You haven't been here long enough to have a rep,
Dang.
but on personality, you're high up on the list.
Oh my god. Thanks, Dr. Sunny.
It's okay. My wife thinks I'm crazy. She knows I'm crazy. Okay. Peptidoglycan. Know about the peptidoglycan, NAMs and NAGs, thick and gram-positive, so it's not the same in all bacteria. Porous, that's why we don't have to worry about water getting through just by channels in the cytoplasmic membrane. That outer membrane has to have porins or transport proteins, so water and water-soluble things can go too. Remember, heat is permanent denaturation, and refrigeration is not. It's just slowing the growth cycle. The difference in all those air things. Obligate means it's got to have it. Obligate anaerobes can't live in oxygen, don't have the means to detoxify it with superoxide dismutase and catalase. Best examples of those are those soil endospores that are anaerobes, like Clostridium botulinum, Clostridium tetanus. They're anaerobic. That's why they only live in anaerobic environments or flourish in that. That's significant. Microaerophiles, a little bit of oxygen. Ones that have gastric ulcer disease or flesh-eating disease or gastric carcinoma, so they definitely affect us. Don't forget the toxic derivatives of oxygen, principally superoxide, but it's all about catalase and superoxide dismutase. What happens when it's too much water or not enough? Pulling water out is the most effective mechanism in bacteria. It causes the plasma membrane to basically burst. Okay, and so it shrinks them and pulls away. Where the cell wall in bacteria, excessive water doesn't make it. Where in our cells, it would cause them to burst in the absence of the cell wall. Facultative grows easily. Fastidious, a lot of growth factors. Run, tumble, run. Light, chemicals, magnetic, air, all of those things are part of it. Heat also, that's why we have ones at the top of the tube, one at the bottom, ones that grow like mesophiles and thermophiles. And then lastly, the phases. Lag phase, log, and I have a minute. Lag phase, log phase, stationary phase, and death phase. Lag phase. Effectively, nothing's growing. It's just accumulating macromolecules, nutrients to get ready to grow. When it grows extraordinarily fast, exponentially. Okay, and two things. Best time to treat it, because they're gobbling anything in their environment, so antibiotics will be gobbled up. Two, primary metabolites, things that we, in science and pharmacologically, that's where we make things like human insulin. Stationary phase, closed system, no more to eat, garbage everywhere, bad environment, endospores can form, metabolites change. Now they're secondary. That's antibiotics, because they're designed as a mechanism for one microorganism to have an advantage over the other. I got antibiotics, you're SOL. Shit out of luck. I didn't say that. Up the proverbial creek without the proverbial paddle. Okay, enough of that nonsense. And death phase, also exponential, but considerably slower, more dying. And it's not about absolutes, it's relative. So stationary because as many are growing as are dying, so it's flat. And that's as much as I can share with you in the elapsed time. Goodbye out there.
Thanks, Dr. Sunny.
Very Todd.